Hello everyone, my name is Estephania Garcia. I am an undergraduate student at UTRGV's College of Health Professions. Today I will be talking about the effects of surface hydrophilicity on pre-osteoblast behavior, a comparison of titanium surfaces. This experiment has been conducted at the Regenerative Medicine Research Laboratory at UTRGV's School of Podiatric Medicine in Harlingen, Texas. To start off, about 30% of our population here in the Rio Grande Valley is diabetic, leading to many cases of impaired wound healing and neuropathy. Charcot's foot, as we can see in Figure 1, is a progressive condition often caused by diabetic neuropathy that weakens the foot and the ankle. The bones become fragile enough to fracture, and over time the foot can change shape. Figure 1 portrays an advanced stage of Charcot's foot that can be treated with surgical fixation. This fixation involves removing the damaged bones and realigning them with screws, rods, and plates. However, this procedure has a 36% complication rate. There's very little research on how different surface treatments can minimize these failures, but what we do know is that surface wettability is very important for long-term implant integration. By surface wettability, we're talking about the surface's ability to either repel or absorb water. In Figure 3, we can see an example of what that would look like and how it can be measured in terms of contact angles. When we see a hydrophobic surface, we'll see a higher contact angle. When we see a hydrophilic surface, we'll be looking at lower contact angles. We hypothesize that by enhancing the hydrophilicity of titanium through a SLA surface treatment, we may also enhance pre-osteoblast cell response, potentially improving clinical scenarios. Throughout this experiment, MC3T3 pre-osteoblast cells were used along with small titanium discs with different surface treatments. To start off, we have the sandblasted treatment, which involves compressed air propelling sand-like beads onto the surface. Smooth titanium, which is what's used in Charcot's fixation today. Acid-etched titanium, which uses a strong acid to leave a different texture on the disc. And SLA, which is a combination of both sandblasted and acid-etched. The cells were seated on these different discs, or different treatment discs, in a 96 and 24 well plate for 3 days. This was followed by an MTSSA to determine cell viability, as well as immunofluorescence imaging to look at the morphology. In Figure 6, we can see the 2 trials conducted for cell viability. Trial 1 involves a normal glucose environment, while Trial 2 involved a high glucose environment. There were no significant differences in either trials, but we believe these outcomes to be preliminary and will be repeating this part of the investigation. Moving on to Figure 7, we can see a comparison of surface wettability and contact angles between one of our control discs and an SLA-treated disc. The control has a much higher contact angle in comparison to the SLA disc. In Figure 8, we can see how there's a significant difference between the two contact angles. And we can see, again in Figure 7, the control will be more hydrophobic surface, and our SLA would be more of a hydrophilic surface. In Figure 9, we're looking at our cells morphologies. We stain the cell cytoplasm with FITC and their nuclei with DAPI. When we look at our negative control, we see how the cells would like to adhere properly, what they would look like when they adhere properly to a surface. And as we go down the panel, we see the difference in morphology when adhering to different surface-treated discs. We conclude that the effects of an SLE treatment highlight beneficial effects in promoting cell proliferation. However, further studies are required to explore the long-term impacts in clinical settings. As I conclude, I'd like to acknowledge the B2B Med program and the Regenerative Research Laboratory for all their help. Thank you, and if there are any questions, feel free to contact me or my PI. Our information can be found in the lower right corner.